Hi class, Miss Sterling here. We're going to do Unit D, Part 3 today, Recombinant DNA. So the key idea of this screencast, we're looking at PLOs D5, 6, and 7, and we are thinking about how we have developed this amazing ability to create in the lab transgenic organisms. So first of all, some vocabulary words that we're going to meet. Recombinant DNA is the technology that is going to be employed to create the organism. So that really is the big idea. The recombinant DNA is what makes a different organism, an organism with new genes inside of it. And then we're going to end off by going back to DNA and RNA and making sure that we understand how they are the same and how they are different. So the compare and contrast idea that we've seen before. So first idea, creating a transgenic organism. So first thing that happens is we have a gene of interest. So we're going to use an example that we've talked about before. We're going to talk about the idea of the gene that codes for insulin. So there is a gene, and that is in the bases of DNA. So it is the code. Codes for a protein. Remembering, it codes for the primary structure of the protein. So in a person that lacks the ability to make insulin, they can then get insulin from an organism such as bacteria who has been given this gene of interest, the gene that codes for insulin. So this gene is inserted into a bacterium. So we add this specific gene. So we're going to pretend it's this little red squiggle. So we've inserted this one gene of interest in the bacteria, and then the bacteria has the ability then to produce the protein that we wanted in the first place, insulin. And this is because DNA's code is universal. So the code in us speaks to our cells the same way that the DNA in the cell of a bacterium does. So the new bacteria that is created is referred to as a transgenic organism. And this is always the outcome of recombinant DNA. It is not the insulin. The insulin is what we want, but the transgenic organism is what we make to get the protein that we want. So putting it all together again, we have a gene donor. So this would be the gene of interest, the code, the piece of DNA that we want. And we are going to get it inside. We're going to isolate it. We're going to use some enzymes to chop it out, remove it. We're going to add it in to a plasmid, and a plasmid is like a little pellet that can be inserted inside of a bacterium. So it's a way to get the DNA, the piece of DNA of interest, inside of the cell. So we get the DNA, here it is, inside the bacterium, and ta-da! Then what happens is that the bacterium expresses, it makes, by adding this gene, it has the ability to express this protein. And that's the whole reason why we put the DNA in the bacterium in the very first place. So recombinant DNA is defined as DNA from one species. So in the example, if we're talking about insulin, it would be DNA from a human being that is inserted or added into the DNA of another species, and this is for human benefit, right? It makes a lot of money. So although there are many good things that have come out of it, it is a technology, it is a money-making operation. So some examples, transgenic crops. So we've all, I think, hopefully heard of GMO, genetically modified crops or organisms are sometimes in the news. So transgenic crops are an example of recombinant DNA technology that either have added nutrition to them, so the crops have added nutrition, or they make the crops more environmentally tolerant, so they can withstand drought, they can withstand salt in the soil. They can withstand heat. An example would be on the right-hand side here. Golden rice was created to add beta-carotene. Vitamin A is deficient in many people in the world. It's a vitamin A deficiency. So as a way to combat this, they created a rice that had inside of it some beta carotene to add to the diet to supplement the people in the world that were lacking beta carotene. So golden rice would be an example. Another example would be transgenic bacteria. Remember that important idea that this technology is creating always a transgenic organism. So another example using bacteria is they've created bacteria that can actually metabolize oil. So they actually ingest it and break it down, and obviously the application of this then would be in oil spills that are not obviously too large. These bacteria can be applied and then they can help to dissipate, get rid of the oil that is causing the environmental disaster. Another example of how transgenic bacteria are used, we already mentioned at the beginning, is to create proteins of interest. So insulin is one. Human growth hormone would be another one that has been used. So insulin, as we read, used to come from slaughtered cows and horses and was expensive and time-consuming. Now we have bacteria that make the insulin for us, and this is cheaper, much more cost-effective. So again, just to recap the idea here, we're coming almost to the end. The pancreas, the cells inside of the pancreas have the code for how to make insulin. So this is coded for in the DNA, in the bases of the DNA. So this is a pretty important idea. So you want to focus on what's important here. So DNA, the bases create a gene, a meaningful sequence. This is cut out, removed, and it's added then into a bacterium, a host bacterium, that then will make the human insulin. So this is the product that we want at the end to use, but to get that, we create a transgenic organism, in this case the bacterium, and we would make lots and lots of these bacteria. Last little topic to hit on, to review, this was Unit D, DNA. We compared DNA to RNA. So although they are quite similar, they have some very specific differences that we should be aware of. We should also be able to recognize these differences in diagram form. So the three key differences are the sugar. In DNA, it is deoxyribose, hence the name DNA. In RNA, it is the sugar ribose. But remember, the sugars look exactly the same, and they are both referred to as pentose sugars. They are both 5-carbon sugars, so they have the little house shape. There is one base different in DNA when compared to RNA. DNA has the base A, T, C, and G. RNA, instead of T, has the base uracil. And so when we meet a molecule, right away, if we see uracil or U in its strand, we know that we're looking at an RNA molecule. Another difference between these two molecules, DNA is look at that, a double helix, and RNA is a single-stranded molecule. Who typed that? Some bad person. So ending off, what are the things that you should be able to do? Well, you should be able to define what recombinant DNA is. You should be able to give some examples and explain them with some meaning, not just transgenic bacteria, right? A little more than that. And then the last one that really is important when we look at Unit E is comparing and contrasting the two nucleic acids, DNA to RNA, and we should know 3 differences there. Hope